Good day once again, my dear students. Welcome to this short discussion about the RNA. In this part of our topic, I will talk about the basic concepts of ribonucleic acid, as well as its structure and vital functions. Like DNA, the ribonucleic acid is an important molecule in regulation and expression of genes. RNA transfers the genetic code from the nucleus to the ribosomes during protein synthesis. Aside from its important roles in protein synthesis, RNA also has other biological functionality, such as communicating cellular signals as well as catalyzing some chemical reactions. Now let me talk about the structure of ribonucleic acid. RNA is found to be single-stranded with ribose as the sugar backbone. With it are the nucleobases adenine, guanine, cytosine, and uracil. Uracil serves as an alternative for thymine as a nucleotide base. However, it is just an unmethylated form of thymine. We have this figure to differentiate DNA from RNA. Ribonucleic acid or RNA is a molecule similar to DNA. However, unlike DNA, RNA is single-stranded. RNA is chemically similar to DNA except that it contains ribose instead of deoxyribose as its sugar and has the nitrogenous base uracil rather than thymine. Thus, each nucleotide along a DNA strand has adenine, guanine, cytosine, or thymine as its base, and each nucleotide along an RNA strand has adenine, guanine, cytosine, or uracil as its base. Now let us talk about the types of RNA. Different types of RNA exist in the cell. We have messenger RNA, ribosomal RNA, and transfer RNA. The importance of each type will be tackled in the succeeding slides. The first type of RNA is the messenger RNA or mRNA. mRNA is a single-stranded RNA molecule that is complementary to one of the DNA strands of a gene. Since DNA is too large to successfully exit the cell's nucleus, the genetic information is carried by the messenger RNA to the cytoplasm. The mRNA is an RNA version of the gene that leaves the cell's nucleus and moves to the cytoplasm where proteins are made. Next to the messenger RNA is the transfer RNA or tRNA. tRNAs are small RNAs with a size of about 80 nucleotides. Its main function is to transfer amino acids to growing polypeptide chains at the ribosomal site of protein synthesis. Each tRNA molecule has two important areas: a trinucleotide region called the anticodon, and a region for attaching a specific amino acid. You will learn more about the tRNAs in our discussion about translation. And then we have the ribosomal RNA or rRNA. rRNAs comprise the majority of RNA found among typical eukaryotic cells. These are the catalytic components of the ribosome. rRNA and proteins combine to form the ribosome, which later on binds with mRNA for protein synthesis. And lastly, we have this special type of RNA. Special in the sense that, unlike the normal single-stranded RNA molecule, this type has two strands. The double-stranded RNA or dsRNA is an RNA with two complementary strands. This is the genetic material found among some viruses, such as the rotaviruses. So this is the end of my short discussion about the RNA. Thank you for watching this video.